So today we will discuss about the metabolism of methionine, cysteine and cysteine. These three are sulfur-containing amino acids. So methionine is an essential, whereas cysteine and cysteines are non-essential amino acids. Cysteine and cysteines are synthesized from two amino acids, methionine and serine. We'll see in the subsequent slides. Cysteine and cysteines are readily interconvertible in the body. Methionine is required for initiation of protein synthesis, and sulfur-containing amino acids are exclusive source of sulfur in the body. So metabolism of methionine we will see in three segments. First is the transmethylation reaction, in which we'll see the conversion of methionine to homocysteine. Second is the synthesis of cysteine. So conversion of demethylated portion of methionine to cysteine and cysteine. Then degradation of cysteine, in which there is a conversion of cysteine to its specialized products. So the first thing which we are discussing here is transmethylation reaction. So the first step in this is the methionine is going to be converted to its active form, that is the S-adenosyl methionine, or in brief SAM. So how it happens? The enzyme is methionine adenosyl transferase, and it's a sort of reaction between the sulfur and the this is the adenosyl moiety, which is a nucleoside. You can see this is the pentose sugar, ribose sugar is there, and this is the base, adenine base. So this is overall adenosine. So it is now attached with sulfur of the methionine. In the presence of enzyme methionine adenosyl transferase, with the loss of water, and 3 ATPs are being required for this conversion. So this reaction is actually the ATP is converted to the 2-pyrophosphate and phosphate. That is equal to loss of 3 high energy bond, and that's how we are telling it. It's the consumption of 3 ATP. Then further, the S-adenosyl methionine, it is all said to like act as a donor of this methyl group, because now methyl group is attached with the sulfur. Sulfur is positively charged, and so this is known as active methyl. This makes the sulfur active, because this is how the sulfur this methyl group is easily labile, and it can be donated to any of the entities wherever it is being required, and sulfur get neutralized by squeezing the bond towards it. So that's how it is the active methyl group. So S-adenosyl methionine is a donor of methyl group in various metabolic reactions and responsible for various biologically important products. We have seen in many previous reactions in tyrosine metabolism and tryptophan metabolism. Like this is the example that methyl transferase is the enzyme which is responsible for transfer of methyl group. So there should be one acceptor in this reaction. So acceptor is going to accept methyl group and convert up into the methylated product. And there can be, I can give one example of norepinephrine to epinephrine conversion. So this is all happen just by acceptance of methyl group. Then this product, this acid adenosyl methionine, after donating the methyl group, it converted up into the S-adenosyl homocysteine. Further, this adenosyl homocysteinase enzyme converts this acid-adenosyl homocysteine into homocysteine, and with the loss of adenosine moiety, this adenosine moiety is being released back. Further, this homocysteine is now converting back to methionine. So this step requires the molecular presence of B12, vitamin B12 and vitamin B9, that is tetrahydrofolic acid, and which is loaded which is loaded with a 5' methyl group. So in 5 position there is a methyl group, and Once homocysteines take back the methyl group, it converts back to methionine. So two important, here comes the role of two important vitamins, vitamin B9 and B12. And the enzyme is homocysteine methyl transferase. And this step is hampered if the any of the component required for the reaction is deficient, or like vitamin B12 is deficient, or the vitamin B9 is deficient, or this enzyme defect can also be the culprit. So in such conditions, there will be no conversion of homocysteine to methionine. So methyl group remain trapped along with the tetrahydrofolate, and this condition is known as methyl trap. So many compounds become functionally active only after the methylation. Methylation protects protein from immediate degradation. It acts just as a protective group in various amino acids. In plants, the SAM is a precursor for synthesis of plant hormone ethylene, which regulates the plant growth and development and is involved in ripening of fruit. So this is all importance of transmethylation reaction and SAM, that is S-adenosyl methionine. So here there is a volley of reactions showing the transmethylation reaction products like guanodoacetic acid converts to creatine by simple acceptance of methyl group once it is methylated with SAM. So here is a role of SAM. Similarly, nicotinamide to N-methyl nicotinamide, norepinephrine to epinephrine, epinephrine to metanephrine, then ethanol amine to choline, carnosine to anserine, acetyl serotonin to melatonin, serine to choline, then histidine to methyl histidine, and lysine to methyl lysine. So transfer RNA, they accept the methyl group and becomes the methylated trans. And these are the initiator transfer RNA of the protein synthesis. So many well-known reactions are compiled here, and here it becomes very significant. It shows the very significant role of SAM or S-adenosylmethionine in methyl transfer reaction. Then homocysteine and heart attack. Homocysteine as an intermediate in synthesis of cysteine from methionine. Its normal level in the plasma is less than 15 micromoles per liter. Increased levels are seen in coronary artery disease. Homocysteine reacts with collagen to produce homocysteine thiolactone, a free radical which thiolates the LDL particles. So they tend to aggregate and increase the tendency of atherogenesis. That's how it makes the person, the excessive amount of homocysteine make the person more prone to atherogenic effect and the cardiovascular diseases. So hyperhomocystinemia in pregnancy increases the risk of neural tube defects in fetus, and treatment lies in the intake of folic acid, vitamin B12 and vitamin B6. They have a beneficial effect in lowering the plasma homocysteine level. So in the previous slide, we have seen how methionine can be converted into the homocysteine via SAM, and it will revert back to methionine. And that cycle, there was no net production of methionine. It was just like a cycle, and homocysteine was just as an intermediate of that cycle. So now homocysteine along with the serine in the presence of cystathionine beta synthase enzyme, which is induced by PLP, pyridoxal phosphate or vitamin B6 active form, and it leads to the synthesis of cystathionine. Further, cystathionine in the presence of enzyme gamma cystathioninase, it converts up into two important fragments. One is the alpha ketobutyrate, which further converts up into the succinyl coenzyme A with the set of few reactions in between. Then cysteine is another fragment, so which is a non-essential amino acid. This is how the methionine can be converted up into the cysteine. And then this is how this slide shows that succinyl coenzyme A, which is an intermediate of Krebs cycle, so it is involved. It may be involved in the gluconeogenesis and the synthesis of glucose. That's how the methionine is glucogenic in nature. Then in this picture, we'll see the cysteine or cysteine. Actually, cysteine is the dimer of cysteine. So any of these combined with homoserine, ammonia is lost because there is enzyme deaminase, and it converts up into alpha ketobutyrate intermediate. That alpha ketobutyrate is converted up into the propionyl coenzyme A, and NAD and coenzyme A are required for this step to happen. Then further propionyl coenzyme A will via methyl malonyl coenzyme A, it converts up into the succinyl coenzyme A, and which is participating in the TCA cycle. So this reaction is very, very important reaction. We have seen in the previous metabolism of tyrosine, as well as we have seen its important generation in the uh, beta oxidation of hot chain fatty acids. So this reaction is very, very important, how the propionyl coenzyme is converted into the biologically useful product, that is the succinyl coenzyme A. So in this reaction, cysteine, once the cysteine is being formed, cysteine can be dimerized to L-cysteine. You can see it here. It's dimerization by the disulfide bond, and the enzyme is cysteine reductase with the loss of two hydrogen. Then in this picture, we'll see cysteine further degrade into the various biologically important or specialized products. So from cysteine to cysteine, from cysteine to cysteine. Cysteine can be decarboxylated to mercaptoethanolamine, which is an important component, important for synthesis of coenzyme A via pantothenic acid. Then cysteine, it can also be converted up into cysteine sulfinate. Cysteine sulfinate desulfate via the enzyme desulfinase. It releases alanine, which is glucogenic, and the sulfide fragment is separated out. This sulfide converted up into the sulfate. This sulfate incorporates in it via ATP into the PAPS, that is the phospho 3 prime phosphoadenosine 5 phospho sulfate. In brief, it is called as PAPS, which is an important source of sulfur or sulfur donor in various act as an intermediate in various metabolic reaction. And then it has important role in synthesis of various mucopolysaccharides, sulfur containing mucopolysaccharides, and detoxification reactions. Further, this cysteine sulfinates can be converted up into the cystic acid, which can convert up into the taurine, and taurine can conjugate with the cholic acid, bile salts. So taurochalates are being formed. Then it can also convert up into the pyruvate, and from pyruvate it turns to glucose, and it can also be responsible for synthesis of fats. And then via sulfurdrase enzyme, it converts up into the pyruvate. So that's all about. This is the degradation in nutshell of the cysteine. So this cysteine can convert it up into the glutathione because it is a tripeptide, gamma glutamyl glycyl cysteine. Gamma glutamyl cysteine glycine is the name of glutathione. Then cysteine can also be involved in mercaptoethanol amine formation, which is important for coenzyme A synthesis. Via cystic acid, it forms the taurine, biogenic amine. Then via sulfide, it forms the PAPS, which is an important donor of sulfur group. Then pyruvate, via pyruvate, it becomes glucogenic. Now we will see the metabolic disorders associated with the sulfur-containing amino acid. They are cystinuria, cystinosis, and homocystinuria. So cystinuria is the most common inborn errors of amino acid metabolism transport. Amino acid transport. So cystinuria is a defective renal absorption of cysteine, ornithine, arginine, and lysine. And the mnemonic is COAL, coal. It is characterized by massive urinary excretion of cysteine, ornithine, arginine, and lysine because all these four amino acids have the common transportation channel. So if it is hampered, so there is no urinary excretion of all these four cysteine, ornithine, arginine, and lysine amino acids that can lead to the formation of renal stone, obstruction, infection, and renal insufficiency. Treatment involve the ingestion of large amount of water. Then cystinosis is a rare but a serious lysosomal disorder. In this particular type, it's caused by the defective carrier that transports cysteine across the lysosomal membrane from lysosome to the cytosol. So due to this defect, there is a severe accumulation of cysteine in the tissue sites and various organs of the body, and accumulation of lysosome in blood and reticuloendothelial system like in spleen, liver, and bones. Cystinosis usually accompanied by generalized amino aciduria. Patient usually die within 10 years due to acute renal failure. Homocystinuria is actually a group of disorders of methionine metabolism characterized by high blood and urinary level of homocysteine and methionine. The four metabolic defects caused due to the homocysteine. It is the homocystinuria type 1, type 2, type 3, and type 4. Type 1 is due to the deficiency or defect of enzyme cystathionine beta synthase. Type 2 is due to the defective synthesis of N5 methyl tetrahydrofolate. And type 3 is due to the deficiency of methyl B12. And type 4 is due to the defective intestinal absorption of vitamin B12. So if we talk about the cystathionine beta synthase, which is responsible for conversion of homocysteine and serine to cysteine via cystathionine. So this enzyme, if it is blocked, so this will lead to the elevated level of homocysteine, and that is the type 1 homocystinuria. Further, while converting homocysteine to methionine, so vitamin B12 or methyl B12 is being required, which converts into vitamin B12. It acts as a donor of methyl group, by which the homocysteine converts it up into the methionine. And further, the vitamin B12 again get methylated and it accepts the methyl group from the N-methyl tetrahydrofolate homocysteine transmethylase enzyme and from the N-methyl tetrahydrofolate. So this methyl is used for methylation of this vitamin B12. So if this enzyme is blocked or there is a defect in this enzyme homocysteine transmethylase, or if there is a deficiency of this tetrahydrofolic acid or vitamin B12, all of these will lead to the no hamperment of or blockage in conversion of homocysteine to methionine. So result will be the increased or elevated level or concentration of homocysteine in the blood and urine. Then further defective absorption of vitamin B12 from the intestine, that will also lead to the deficiency of vitamin B12 in the body. So that will hamper this step of conversion of methionine to homocysteine. So both these vitamin B12 and B9 are integral components related to the methionine metabolism. Without this vitamin, methionine will not be able to convert to homocysteine will not be converted or revert back to methionine. So, and the next cause is deficiency of this enzyme N5 and 10 methylene tetrahydrofolate reductase, that is responsible for conversion of N5 and 10 methylene tetrahydrofolate to N5 methyl tetrahydrofolate. So this is the defect in this enzyme is called homocystinuria type 2. So these were the various causes of different types of homocystinuria.